So today we're joined by Professor Vladimir Torchilin, who is Director of Center of Pharmaceutical Biotechnology and Nanomedicine at Northeastern University. Thank you for being here. And
The pleasure is mine.
how are you?
I cannot complain. Would help anyway.
Good. So, I would like to start with your background, as I know you studied chemistry at Moscow State University or MGU, and you specialized in physiologically active polymers. So, can you explain how it all started, and how did you transition to United States, and how was your journey?
It's a good question. So, yes, correct. I am graduate of Moscow State Chemical Department, and I defended my PhD there, and I defended my Doctor of Science there. Uh, so, good question. How I left. So, we have to touch politics anyway. So, because living in Soviet Union, I hated it from the childhood. I hated it even more because I had to comply with many of the requirements. So science was more or less safe area in this sense because as little politics as possible. But still, I had to comply with many things, including in order to become laboratory chief, I had to be the party member. So I was thinking about any opportunity to live with, and the simplest was because of my Jewish heritage, I could immigrate to Israel. However, my wife, who is ethnic Russian, and she is the only daughter, so she was afraid to leave her parents because that time, if you are immigrating, the contacts with Russia became virtually impossible. When the better time came, like at Gorbachev time, when immigration was still a problem, but not as much problem. And besides, people were free to travel, so even those who immigrated still could meet their relatives somewhere in Europe. But by that time, I had another kind of difficulties because in 1982, in a group of other scientists, I got Lenin Prize in science and technology, and people with highest national award, they had far too many troubles in immigration because the state didn't want us to leave. Certainly, it makes the bad reputation. If the most advanced people are leaving, the most awarded people are leaving, what does it mean? So, and I used an option when I was with one of the conferences abroad. I just refused to come back and ask for political asylum.
Which country was it?
It was 1990.
And in which country?
Say it again?
In which country did you stay?
In the U.S. However, my lawyer uh, gave me very good advice not ask a political asylum, which was pretty difficult because it still was kind of a Gorbachev time, but it was attempt of military revolt in Soviet Union, so it could work or it couldn't work. And he suggested me to apply for permission to stay as a ally of outstanding abilities, which which I certainly could meet with my reputation, international reputation in science. So this how it worked, and I started my life there in 1991. I got my permanent position with Harvard Medical School. By that time, my wife immigrated in a more difficult way, being on a private invitation in Europe, so that he traveled to the United States, then we joined. We actually started from scratch because we both came with virtually nothing. Just the only luggage we have, it was 1 week travel. But that was fun. So I stayed for almost 10 years with Harvard Medical School, and then I was invited to apply for a position of department chair at another good university University in Boston, Northeastern University. And in late 90s, can't tell exactly when, but late 90s, I joined them, and since that time I'm staying with them, and I never regretted about my move. So I like this university. University is very good now. Oh,
And what
that's it.
challenges did you encounter in this
Close to
process?
mic. I don't hear you.
What challenges did you encounter in this process? Maybe like cultural barriers, language barriers.
No, I spoke fluent English because my mother, who knew that I wouldn't stay in that country, she trained me in English from the childhood. I had the English teacher, private teacher, so I was pretty fluent in English. Besides, I was a part of collaboration between U.S. and USSR in the area of basic cardiology, so cell biology, biochemistry, and as such, we had a permanent exchange with American labs. So we were got quite a few people from our institute were visiting United States as exchange scientists, and Americans were coming to our labs. So I was already several times working in one of the lab of Mass General, which is part of Harvard Medical School. So I knew pretty well where I'm going. Certainly, it was like a big step down because in Moscow I was like nationally awarded, nationally recognized professor and department chair, and here I started like almost like a junior researcher, but that's fine. So it was like a second chance, and within like a couple of years I was back on track with my initially small group, eventually very large group at the peak of my funding. So I had a lab of about 40 people. So no, I didn't have any troubles.
And were there any differences in like scientific culture in Soviet Union and in the United States, like the process you had to go through?
In general, yes. In our particular case, no, because after the university, I joined the new venture, which only appeared at that time. It was the National Institute of Cardiology. I believe this was the title at that time, of Academy of Medical Sciences. And its head, academician Chazov, who simultaneously at different time points was a Minister of Health and the head of Kremlin Hospital, and very influential and very smart person. I know that he was outstanding physician, which I cannot just judge myself, but it looks like he was. But he certainly was a great organizer and he had a very good vision for how science should develop. So with approval from the very top, he has some connection, probably as a Kremlin hospital director. He got a permission to build an institute which function and behave like a very good American institution, a very good international institution. Certainly, it was very different from the rest of the country, from the majority of the country. And one of the biggest differences was like in Soviet Union, and I believe still in Russia. I don't know much what's going on there now. Still, one person or very small group of people can control the whole area. So it was like the very frequent case, like the big academician simultaneously was the director of a head institute of this area, was editor-in-chief of the main journal in this area, and naturally he or she controlled more frequently he. She was very rare. I know only one, Bechterev in Leningrad. He could control actually the whole area, which is very different from United States. It doesn't matter how influential you are here, there's still maybe not as highly influential, but very similar ranks people in other universities, and there is no chance that anybody could direct or control the whole area. This is a big difference in how science is arranged. But as far as I know, now in Russia they have the same. They have grant system, and it doesn't help them to be a normal country, but it's their problem. not my problem anymore.
So you studied the psychologically active polymers. What are they, and where are they used? Where do you use them?
Uh, so okay, I believe that you and the listeners they know what polymers are.
Yes, but
Polymers are like a chemical structure of a very high molecular weight, which can be prepared from variety of small molecules and can express, the huge number of different properties, which can be replacing metals or just whatever. So everyone knows with plastics. Less people know that we, as natural creatures, are built from other polymers, which are proteins. So the proteins are biological proteins. Polymers are chemical products. So, but people always had a dream to build such polymers which could mimic biological properties. And this was a problem I was involved in a very interesting group at Moscow State. So, among the natural proteins, there are certain proteins which express enzymatic properties. So the polymers which catalyze or inhibit biological processes. So, and when the structure of those proteins was understood, more or less, the idea was quite natural to repeat them by chemical synthesis, to try to synthesize polymer which would behave like natural proteins. And this might be like an endless area of application. You can just use it for blood replacement, for variety of treatments. And this is how I got involved in these physiologically active proteins, and my PhD was about that. Now the next step is just to natural proteins, such as natural enzymes. And when I was invited to join this cardiology center, they have like a very interesting task, which was related to the protein enzymes used as drugs, as treatment system. For example, to dissolve thrombi, to treat like myocardial infarct or pulmonary artery thromboembolism. The problem with those enzymes was that they were unstable, they provoke immune responses in the body, and the task put in front of me and my group was how to preserve good properties of those enzymes, minimizing their harmful side effects. And you see,
this
it
is
was quite
happening in
logical.
Russia, right?
Polymers, then physiological polymers, then enzyme, then how to make the delivery system for enzymes.
This is happening in Russia, right?
Yes. So, and this is how I came to this area of drugs and physiological actives and ways for their modification and delivery. And after that, I already was in this area all the time, just switching a little bit my targets from cardiology to oncology or to some other areas.
And how did you continue in the United States? Did you continue with the same physiologically active polymers, or did you move to other topics as well?
The step was not as much with physiological actives themselves as with means for their delivery. Because for many physiological actives to modify them in desirable way, it's not a big problem after you understand some mechanism. So it's pretty simple. But after that, you have like a very simple problem. For example, you would like to treat thrombi, to dissolve thrombi, fine. You have enzyme to do it, you modify it, it behaves exactly the way you like. But in order to reach thrombi, which is a very small piece in your circulation system, you have to inject huge quantity of enzyme to create active therapeutic concentration in the area of thrombus. So it should be everywhere. Enzymes are very expensive, very unstable. So you have infused them instead of single injection, which requires like clinical condition to make an infusion system, and it costs a lot. But now imagine that if you can modify enzyme in such a way that it will go only to the area of thrombi, not just being taken anywhere else, you can save a lot in terms of time and money and efficacy. So this is a bridge between making modified physiological actives and making system for delivery of these physiological actives.
And how do you make enzymes go to the specific thrombi or a specific location that you desire?
Oh, it's probably like a very good example to use. It's to explain you, for example, what we got this national award for. So that time, I mean, a lot of change that time. We are talking about studies which have been done 50 years back in mid-70s, 1970s. Enzymes used that time for dissolving thrombi were produced separated from a microbial system of streptococci. And streptococci, we all are infected with streptococcus. And we all have antibodies against streptococcus in our body. So when you make the enzyme purified from streptococcal culture and inject it into human body, the terrible immune response takes place. So enzyme is not only unstable, it also provokes strong allergy. First administration usually is okay, but second administration can result in whatever complication you can think about, including anaphylactic shock, which might result in death. So ideally, you have to have the enzyme which is stable and not immunogenic. So for doing that, you first have to understand how enzyme inactivates and how can you prevent it. So it created the whole big area of enzyme inactivation and stabilization. And our input in this area was really very big. We published a series of papers in the best biochemical journals of that time. So after we understood it, so it became clear how we can stabilize it. We can stabilize it by attaching to synthetic polymer. It's like when you have a ship attached to the pier with few ropes, it stays solidly. And this is exactly what you can do. If you can attach protein to artificial rigid polymer with several bonds, the protein cannot unfold anymore. So you stabilize it. Second was to find out what causes immune reaction, what part of this protein. After you can find it, and important is that active part of the protein and immunogenic part of the protein are different parts. Then you can specifically modify. Actually, it's much more simple that I'm explaining. You have to modify the immunogenic part so you cover it from the recognition by body immune system. Now you get a polymer which is stabilized and which is non-immunogenic, but still active. And this is what we did. We developed the theory of enzyme stabilization. For this particular enzyme, which was used, streptokinase, which was used as a therapeutic mean, we found the way to stabilize it based on our principle and make it non-immunogenic. So it became like a new product with much better efficacy, and this product, to the best of my knowledge, was produced in Soviet Union till its collapse, because the main production unit was in city of Riga, which became like capital of independent Latvian state, and they broke all the contracts with the rest of former Soviet Union. But by that time already new enzymes appear on the market, so it was no need to produce this one. But this one was used for quite a time, so we actually, this explains you, I believe, how it works.
Yes, and so this type of approach was completely innovative, right? So you were the
That
first
time,
guys to do it.
that time, yes.
And so, like, this is how does this convert to like personalized medicine, and how can you use polymers to get to that?
Uh, First of all, do we have clear understanding what does it mean, personalized medicine? I don't. I don't know what does it mean.
Like, there were this big talks about like producing medicine that is specifically targeting hosts' diseases. So, like, for example, if a person has cancer, it would target specifically cancer cells of this person with this DNA and like this specific.
Yeah, I wouldn't call it personalized medicine because the same principle would be applicable for any patient. Like the same enzyme is used for any patient with thrombotic disease. Same stuff is about anti-cancer drug. Here is a small trick. Uh, I believe when people are talking about personalized medicine, they mean the medicine adjusted for this given patient. Actually, I can imagine this only in very specific cases, like, for example, the treatment of genetic disease. Then you really deal with a certain patient with his or her own genetic problems. And like the gene editing might help in this case. So this is personal medicine. I have nothing to do with. I am dealing with delivery of RNA and related substances, but not on the level of personal treatment. Another example when personalized medicine can work is cancer, at least some types of cancer. This is a very interesting phenomenon. You know what biopsy is. Biopsy is like a piece of sample of tissue from the tumor of given patient. So a pathologist look this piece under microscope. It's not good to show on our cell, but let's assume that we have two patients. We don't see them, but they're on both sides of our screen. They have the same tumor. And pathologist sees the same type of cells under microscope. And he says we have this, it's like sarcoma, or it is blastoma, or anything like that. And then you start treating them with a drug which is approved for this type of disease. And you see that response is very different. And this is still mystery, one of the many mysteries of cancer. Why the same cell in different patients behave differently? This is where personalized medicine comes. There are a few companies in the States which are doing the following. They are getting fresh biopsies from the nearest hospital because everything should work very, very fast. And for few hours, those biopsies, they retain all the properties of the initial tumor. Then they divide biopsy in several pieces and treat each piece with a different drug. And they find which is the best drug for this very patient. Because it might happen that the same tumor in patient on the left is better treated with drug A, while patient on this side with drug B. So, and certainly they just tell to hospital. It's certainly everything is encoded, and they tell, look, the person number A55 should be treated with, doxorubicin. The person with C83 should be treated with paclitaxel. This is another type of personalized medicine. But what I'm doing is a very general. It's a very general principles. How drugs could be delivered with high efficacy in certain points of disease or certain points of the body. And this should be applicable for a broad cohort of patients.
Mm-hmm. And since we're talking about cancer, so what is siRNA? How does it work in treating cancer? Is it applicable to all cancers? And is gene silencing the future of cancer treatment, in your opinion?
Well, again, RNA it's a very broad stuff. There are quite a few RNAs.
I said siRNA.
Yeah, but
Yeah.
siRNA is a very special case, and the case I am mostly interested in. The idea of a siRNA is very simple. A siRNA is, roughly speaking, without details, is a small fragment of nucleotide sequence which can inhibit production of a certain protein by blocking transfer of signal uh, to start production of a. Why it's so important? Because the main problem with cancer cells is that they are quite resistant to any type of therapy. This is where the term multi-drug resistant came. So there are more and more multi-drug resistant cancers. Multi-drug resistant doesn't appear from nowhere. It appears as a response of cancer cells for previous therapies. And the main mechanism of cancer drug resistance are that there are certain proteins produced which block the signal which requires cell death. And these proteins are known. They are different in different tumors, so I wouldn't go in names because it wouldn't tell you anything, but there are proteins which prevent cancer cell death. So if you can inhibit production of these proteins, you can facilitate cancer cell death. So the same dosage of the drug which was not efficient before that becomes efficient. And this is what I'm very much interested in, and this is how it can work. So you combine traditional drug therapy with siRNA therapy. So by doing that, you first block ability of cell to defend itself, and when this affinity is blocked, your drug just kill it like it would kill non-resistant cell.
And is it working for all types of cancers, or is it more effective for a particular type of cancer?
Well, it's hard to say. Just people certainly concentrate on the most deadly cancer, like brain tumors or pancreatic tumors, like the prostate cancer or breast cancer, are pretty well controlled with traditional means. But if the system is developed, and again, this is a step to personalized medicine. It might happen that in two different patients, the resistance is defined by two different proteins. So there are two genes exist. Multi-drug resistant gene 1, multi-drug resistant gene 2. They both produce different proteins. They both request production of different proteins. So different siRNA could be used in different patients with the same cancer.
And so you mentioned multi-drug resistant cancers, and so if it happens because of previous treatments of this particular cancer, if that's how they are resistant to this drug, how is it spread? Since I can't contaminate somebody else with cancer, how
No,
does this cancer become drug resistant, even though I didn't pass it to anyone?
no. It's first, it could be intrinsic, that certain cancer cells might grow just having already overproduction. On the other hand, we might be talking about the patient who was treated, got into remission, but eventually the tumor reappeared, and this tumor would be more resistant to the treatment than the initial tumor was. So there are a lot of cases when multi-drug resistance can show itself.
And now for the conspiracy, there are a lot of people who say that cancer medicine exists, but it's economically not good for the pharma companies to use it, and that's why they prolong it with chemotherapy. What's your take on that?
Not much. I doubt, actually. Saying that, you have to assume that all millions of people who are involved in the activity of pharmaceutical companies do not suspect about that, and somebody behind their back. I mean, just be realistic. It's impossible. Certainly, it's a problem of profit, and pharmacology is pretty costly area. But if you think that all prices go up, what does it mean to get a drug from a laboratory bench to the patient bed? Before, even if you got just everything beautiful, I mean, cell data, animal data, it still doesn't guarantee that human data would be as good. Because even tumor grown in rodents, because rats are pretty close to humans, I mean, in many senses, in my opinion. So, it still doesn't mean that it will work anyway. So the best is like monkeys, but monkeys are extremely expensive. So, but even after that, you still have to go to phase 1, phase 2, and phase 3 of clinical studies. Phase 1 is just toxicity. You have to prove that your drug, your system, just whatever is injected into a patient is safe. Nobody is asking if it works. Just has to be safe. You're saying that this dose should treat disease. I suspect, I believe. They say, okay, show that this dose is not toxic. And this is all you have to show in phase 1. Now, phase 2, you already have to show, if you're allowed to go to phase 2, you already have to show that it works against the disease you said, better than like previous treatments, which requires involvement of many patient clinics. It costs huge money. Who pays it? Company. So the company has after that go to phase 3, which means that few huge hospitals should be involved with already a few thousand of patients. Now, can you imagine how expensive this is? So the company has somehow to return this money, because this is why they invested in this drug. They believe that they'll be able to return from sales. Certainly, they add this additional cost, and they making big money, because sometimes this additional cost might be, I mean, much higher than the expenses. For example, like the companies who invented vaccines, mRNA vaccines against COVID, they make billions. Did they deserve it? I doubt, but this is how free market system works.
And so for the COVID vaccines, what was so special about them? So, like, how did the old-fashioned, more widely used vaccines work? And what's the difference with this new mRNA approach?
It's an interesting question. First of all, I'm not sure that all vaccines like the anti-flu vaccines wouldn't work at all. From the very beginning, we have been told that nothing works, which couldn't be true. Because my experience shows that you can find solution of any problem if you want to. In this particular case, the idea was really good, that if you can find like a certain very specific protein which present in the virus of COVID, then you can block its entry into cells, into human cells. And this is what these vaccines do. They were producing antibodies against this S protein to prevent it in infecting human cells. Was it the only possible way? Again, I doubt. But this was a way which, in a sense, was successful. Now, the more general question is, was it really so necessary to do this absolutely required vaccination? It's an open question for me, and none of the arguments which I heard from both sides were convincing. So I maintain neutrality in this issue. So.
And
simultaneously, I got vaccinated as requested, and I got COVID 3 times. So,
yeah, so like that's what I'm interested in. Were these vaccines effective? Like, did they work? Because from what I observed,
you can find only statistic data, because you cannot build experiment on yourself. I mean, you cannot be simultaneously with vaccine and without vaccine.
Mm-hmm.
So what was done, and what was most convincing for me in this part, then if we are talking about people, age people, let's say above 60, and if you are talking about people who got lung complications, the percentage of death among non-vaccinated people was much higher than among vaccinated people. So it's pretty good argument. it tells that even if vaccine doesn't help to prevent disease completely, it increases body's affinity to overcome the disease, which is also good. In younger people, statistic was not that convincing. It still kind of shows it, but I mean, there are some data, as I said.
And so expectations were that these vaccines would at first prevent the spread of COVID. Why didn't they? So, like, what's so special about COVID that these vaccines couldn't stop the spread of it?
Actually, nothing. Nothing. I don't know if you undergo vaccination against flu, which is done every year.
Yeah.
No, does it prevent flu? No, it doesn't prevent flu. I mean, there's still a lot of cases of flu. So same with COVID. First of all, we don't know what would happen without vaccine. But second, you see now we are much more quiet about that. The COVID is still there. I mean, it didn't go anywhere. It would again go in waves, like a flu does. But now we're taking this more quiet, more calm view. So there's no reason for panic. And it was a good lesson that if the next epidemics would appear, and it certainly would, because this is how the nature works, I believe that it would be met much more reasonably.
And how dangerous was COVID really?
How?
How dangerous was it really? Like,
Good question. Scott question. No, not for me. I know that a lot of people died of COVID because of many unexpected complications. For example, like lung dysfunction, which is not that frequent at flu, was much more frequent at COVID, and just many hospitals did not have enough facilities to treat patients like that. So, but I cannot say more than that.
and so some, like, there are these theories that vaccination caused later complications, some tumors, some thrombi, like all of these things. Is it real, and do we have evidence for that?
I never saw any convincing data, and people talk. I mean, people talk. This is what people do.
So no scientific data about convincing.
No convincing data. Let me put it this way: not every data published in scientific journal is still convincing.
Can you explain liposomes and micelles?
Micelles, yeah.
Micelles. Now, what's their role in drug delivery, and how do they work? And, like, I think that's also one of your specializations.
It's very difficult to explain without slides and little, but actually it's very simple. In all drug delivery systems, the reasons for using carriers, pharmaceutical carriers, it's whatever drug is included in. There are just three reasons for that. You can read them in hundreds of papers, but still it comes to three reasons. First, you can protect drug from the body, because many drugs are very unstable. We already talked about that, like enzymes, like nucleotides and so forth. They are degraded in the body very fast. So you need to preserve drug till the moment it comes to required site. So you need a carrier to preserve drug from the body. On the other hand, you need to protect body from the drug, because let's say anti-cancer agents, they not only kill cancer cells, they damage a lot of normal cells, which we certainly don't like. We all know consequences of anti-cancer therapy. And the third is, even if you have drug which is stable and doesn't provoke any harm effects, it might have what is called unfavorable pharmacokinetics. For example, it's cleared from the body very fast, or it doesn't go to the area you want it to go, or it doesn't enter cells if you would like it to enter inside cells. So you are developing carrier which can solve all these problems. So what liposomes are? Liposomes are just lipid bubbles. Lipids are like molecules of which all cell membranes are built, so it's very physiological molecules. Under certain conditions, those lipids, they form very small bubbles. Really small, it's nano size, so 100 nanometers, 200 nanometers, which can be injected into body without obstructing capillaries. But since this is a bubble, it has internal space. And into this internal space, you can put your water-soluble drug because it's aqueous medium inside. Now, when it goes into the body, and if you can make this particle to stay in the blood long enough, and there are means to do it. The means to do it, and actually, I was one of the people who first discovered those means. It was in late 80s.
What are those means? Can you briefly describe?
There are certain polymers which can be attached on the surface of liposome. And again, chemistry behind it is very simple. And these polymers, they behave in a very unusual way. They create like a pseudo cover from the surface, which means that a very small concentration of this polymer can form kind of a gel, like aqueous gel. And this gel does not allow immune cells to recognize this liposome as foreign particle and remove it from the body. So it travels unrecognized, so-called long circulating or stealth liposomes. Which is really like a stealth technology because this is what stealth is doing. Now you are coming to a very interesting point that it was discovered, and the late Hiroshi Maeda from Japan was like father of this discovery. He discovered it for tumors, but eventually it was confirmed for many other cases that in experimental animals always, in humans to some extent, the very interesting phenomenon exists, that under hypoxic condition, it's insufficient blood supply. The blood vessels become perforated. The holes appear in blood vessel wall. And through this hole, small particles can extravasate. They can leave blood vessel and go into tissue. And this is a way they can accumulate eventually in the site of disease, such as tumor or such as infarct. Because they are still made of biological molecules, they gradually just dissolve or fall apart, and drug is released, creating high local concentration. That's exactly what we want. The micelles are very similar system, but they are designed to transfer non-water soluble drugs, because micelles are built in such a way that instead of aqueous interior, they have lipid interior, and lipid can dissolve water insoluble compounds. So the rest is about the same. Just the only difference is micelles are a bit smaller in the size. So it's actually pretty simple, especially for chemists.
It's simple when you explain it.
Oh, yeah, that's good.
And what do you think are some of the biggest discoveries in drug delivery technologies in recent years, and what do you think, where is the future going to lead us?
Unfortunately, how it is built, that the distance between a lab bench and drug really produced is very high. What is going now? That liposomes have been discovered by Sir Alec Bentham in 1963. Yeah, so it was 60 years back. They were recognized as drug delivery system almost instantly. The first papers how to use them for drug delivery appeared in 1965. The first liposomal drug appeared on the market in mid-90s. So it took 30 years before appearing of the first product. With micelles, the story is almost the same. The people start working with micelle as delivery system in early 80s. The first micelle-based drug appeared on the market in early 2000. So as of today, we have it's very difficult to calculate exactly because different countries have different approval systems. So the drug which is used in one country might not be allowed in another country and so forth. But altogether, roughly speaking, on today's market, we have about 30 products based on use of liposomes, including cosmetics products. And few products are 3 to 5, like using micelles. So which means that industry is approximately 30 years behind what was done by scientists. So what we are doing today probably will require like another, it accelerates a bit, let's say, but it requires another 10 to 15 years to come to the market. What will be the leader of this coming? Very difficult to predict because a lot depends on the business surrounding, on what company is most successful. So there are quite a few companies which actively develop vaccines based on delivery system of mRNA. There are companies which develop liposomal delivery for brain cancer. So there are so many biotechnology companies developing this type of next generation product because as of now we have non-targeted but using carrier drug. The next step probably would be to have targeted. The step after next would be to have already targeted not just to certain type of disease, but to certain type of cells, or even more, inside cells to a certain type of intracellular organelles. So this is how it
So,
goes.
how far away do you think that is? How many years do we need to get to that?
10 to 15. You will see it, I will not.
I hope you will too.
No.
And are the pharma regulations on pharma companies, government regulations, are they hindering the process? Are they making it difficult to like over complicating the process, or is it
No,
necessary
I
to
don't
have?
believe so. I believe the system is very smart and very reasonable. It's how to say it's very rigid, but it's better to be overcautious. I mean, everyone remembers the example from mid last century with thalidomide, which was broadly used and taken as a painkiller all over Europe. And 9 months after that, the crippled kids were born. So, and this thalidomide story just really changed the whole approach how to improve drugs. Because by that time, nobody even checked the drug for like teratogenicity. So, which is how it can act on the next generation. Now, all those tests are absolutely required, unless we are dealing with severe diseases which are absolutely deadly, and usually those very deadly diseases are typical not for very big number of patients. So there is a system of approval of so-called orphan drugs. So for these special cases, the regulations might be lifted to a certain extent. For like more traditional drugs like painkillers, thrombolytic enzyme, anti-cancer drug, I believe that all current requirements are absolutely reasonable, despite they're very difficult, they're very expensive. But without that, we might have problems. We still have problems, but now those problems are manageable. If, for example, you have a drug which can treat hundreds of thousands patients, and only one is killed because of side effects, then you have to seriously think, does it deserve to be on the market or it is not so? And this is decision comes in negotiation between companies and Food and Drug Administration. So no, I believe that the system is rigid, but very reasonable, and actually it protects the health of the population.
But since like this is a very complex and costly process, doesn't it cause the medicine on the market to be way more expensive than it should be, and thus
Who
not?
said who said how it should be?
Well, it's if it's not accessible to people because of the cost, and this cost is caused by this overcomplicated or not overcomplicated, just complicated process. Is it
Usually,
worth it?
usually those drugs do not go on the market because, again, this is an issue of negotiation between pharmaceutical companies and insurance companies. So, would insurance would be ready to pay for these drugs? And certainly, it doesn't. Like, still, a lot of very expensive drugs are available because insurance companies cover them. The good example is Ozempic. Ozempic, which is now broadly used in many cases, started as an anti-diabetic drug. Eventually, it has some other applications. It's very effective. It's really outstanding discovery. The single dose cost, if you buy it like without insurance, more than $1,000. With insurance, it costs like, I don't know, depending on the type of insurance, anywhere between $20 and $50. However, I believe that if you will make this outstanding drug, which will cost like $1 million per dose, it would never go on the market. Those drugs like that would never appear.
And what do you think of Ozempic? Is it should it be used as a weight loss drug, or should it be kept as a
No, it's
diabetes
specifically anti-diabetic drug. The weight loss is actually it's not a weight loss; it's a control of appetite center. And this is not my area, but I'm following what people publish about it. They believe, I mean, the people who study Ozempic, they believe that actually it can act on many addiction-related brain centers. So it can be used for treatment of different addictions, starting from narcotic addiction to alcohol addiction, even for gambling. I mean, it's a very interesting molecule. So.
And so, but it shouldn't be used right now for that purposes, right? Because of lack of evidence, or people should use it.
No, I don't believe that somebody specifically uses it, but this is what people who study it publish. So it might be like one of the future uses.
Mm-hmm.
Like, Viagra appeared from like a very different application area. So Ozempic, I believe Ozempic has a big future.
So let's move on to your works in literature. So you're not only a scientist, but you're a writer as well. What brought you to this? How did you start writing? And I didn't get a chance to read any of your works. So which ones would you recommend me to read?
Oh, it depends what you can buy, because I have quite a few books published in Russian and one book published in English. One which is in English is available on, and actually one in Russian is still available on Amazon.
Mm-hmm.
So
And are other
now
books?
I'm writing from my first publications back in Soviet Union back to 80s. So, and certainly I always had an advantage, since writing did not what fed me. So actually I was getting my income from doing science. I was pretty independent what I can write. So this is why I was very little published in Soviet Union when it was Soviet Union. I was pretty well published there after 90s. So even after my immigration, I was sending my things to I always call it to Russian journals and Russian publishing houses. They published my book. As I said, as of today, I have 9 books published. The first one was, I believe, somewhere early 90s, and the last was just before the war.
When did you discover you wanted to start writing? Did you hear me?
Now do.
When did you discover you wanted to start writing, and is writing a passion, or is chemistry a passion to you?
No, I believe that both are. So it's certainly chemistry can live without writing, and writing can live without chemistry, but it both works. So in the writing, again, since I'm not, how to call it, I am not the writer who depends on his writing. So I might write, the professional writer for whom this is the only thing to do. He has to write, does he want it or not? So, and this is a matter of responsibility, and like poor Dostoevsky, he was dictating his stuff continuously because he needs to be published all the time because he always needs money. The people like me who are not getting income from writing can wait till the moment when the desire to write something becomes really intolerable, and you have to write it down just to get rid of it and feel good again. So I think that there is a difference.
And how do you have time to write and do the work that you do?
We have 24 hours a day. It's a lot of time.
How much books have you written? How many books are there?
As of today, I have 9 books published. Some of them overlap a little bit naturally because those are mainly selection of novellas and short stories. But if you put everything together, it would be like 3, 4 good volumes.
And you also write poetry, right?
A little bit less than prose, but yes.
And are all of the books available online or just some?
Fairly, I don't know. You can easily. I don't know what book buying center are you using, because I'm sure that on Russian book center you still can find at least some of those. On Amazon, I know for sure that there is an English book and the last Russian book. About others, I can hardly say. But probably if you want, you can find it.
And just like recommend a book, which one should we read? Which is like the one that you like the most?
No, probably like 2 books they contain. One is called Labuch. You know what Labuch is?
No.
Do you have fluent Russian?
Yes.
I doubt. Labuch is a very traditional name for the jazz musician. Actually, initially, it was used to name musicians who are making money by playing at funerals and stuff like that. Eventually, it was applicable to musicians who play in jazz bands, not big, not official, just paying for money, just in nightclubs, whatever. So, and the verb labat means play in this condition. So the labuch is like a jazz musician playing for money in small groups. And another book is Dom na Morosейке, House on Morosейka Street. So,
okay. Thank you for being here. It was a pleasure talking to you. Thank you.
Thank you, Oto. Just good luck with you. Good luck with your endeavor.
Thank you very much.
Take care.